Hello, I'm Axel Nohturfft. To study the coordination of different lipid synthesis pathways during membrane biogenesis, it's useful to have an experimental system where membrane biogenesis occurs rapidly and inducibly. We found that phagocytosis of latex beads is particularly useful for such studies because cells very quickly synthesize new membranes to replenish those that were lost as wrapping material early during the engulfment process. In this video, my colleague Shaochong Zhang will demonstrate procedures we use to study phagocytosis-induced changes in gene expression using a Luciferase-based reporter gene assay approach. Where's Shaochong?
Hi, I'm Shaochong Zhang from the laboratory of Axel Nohturfft in the Department of Molecular and Cellular Biology at Harvard University. Today, I will show you how to use the reporter gene assay to analyze the regulation of genes involved in membrane biogenesis after phagocytosis. So let's get started. For this procedure, we use cultures of human embryonic kidney 293 cells. These cells are grown in 10 cm culture dishes in medium A, which consists of tubercles, modified Eagles medium or DMEM, supplemented with 10% fetal bovine serum or FBS and a cocktail of antibiotics. Using a standard trypsinization technique, the cells are detached from the bottom of the dish